Manju S. Kumar, Assistant Professor, Department of Biosciences, to deliver the welcome address. Over to you, ma'am.
Thank you, Ashley. A very ma'am, good morning to all. Welcome to the third session of our international webinar series, Biogen 2020, Surviving the Viral Pandemic, a Disease Tolerance Perspective. Today's topic is recent progress on neutralizing antibodies and small molecules drug discovery against SARS-CoV-2. We are happy and honored with the presence of Dr. Kathireshan Natrajan, NIH Fellow, Physiology and Biomedical Engineering, Mayo Clinic, Rochester, USA, for the presentation of this topic. On behalf of the Department of Biosciences and my behalf, I extend a hearty welcome to Dr. Kathireshan Natrajan to this webinar. Then I extend an amiable welcome to all the dignitaries, faculty members, students, and all other participants. Hope the talk will captivate all. Thank you.
Thank you, ma'am. Our speaker for today is Dr. Kathireshan Natrajan, and he will be conveying his enlightening insight on the recent progresses on drug discoveries against SARS-CoV-2. Now, I invite Srimati Fabi K. John, Assistant Professor of the Department of Biosciences, to introduce the speaker.
Thank you, Ashley. Very good morning to one and all. It's my great privilege to introduce our guest of honor and speaker of today's section, Dr. Kathireshan Natrajan, NIH Fellow, Physiology and Biomedical Engineering, Mayo Clinic, Rochester, USA. Dr. Kathireshan has nearly 15 years of experience in research in various fields of biological sciences. He started his career as a lecturer in biotechnology, J.J. College of Arts and Science, Pudukkottai, India. His passion for research led him to Indian Institute of Technology, Madras, India, for pursuing PhD on computational biophysics. His area of interest includes subjects like computational biophysics, molecular biology, immunology, RNA technology, and bioinformatics. Dr. Kathireshan worked as a Junior Research Fellow in Cancer Biology Lab of Rajiv Gandhi Center for Biotechnology, Kerala, India, and as Research Associate in Computational Material Sciences in JNCSR, Bangalore, India. He worked as a postdoctoral fellow in Material Sciences and He has published over 13 research papers and presented scientific papers and posters at national and international conferences. Presently, he is working as NIH Fellow in Physiology and Biomedical Engineering, Mayo Clinic, USA. Today, he will be delivering a talk on the title, Recent Progress on Neutralizing Antibodies and Small Molecules Drug Discovery Against SARS-CoV-2. I think the entitled talk will be very relevant to this pandemic situation. Thank you. Over to you, Ashley.
Thank you, ma'am. Now we can begin our technical session. As usual, our session will start with the presentation of the speaker, followed by a question and answer session towards the end. We request our participants to use the chat box facility to raise your queries, if any. The posted queries will be posed to the speaker at the end of the session after screening. Once again, kindly make sure that your audio is muted and video is turned off to avoid any disturbances during the presentation. Dr. Kathireshan, now the dais is yours.
Thank you for the warm welcome. First, I would like to thank the management and the staffs of Sri Sankara College, especially Fabi K. John. She is my student in JJ College. She is one of the brilliant students. Nice to see her after a very long time. Okay, thank you so much for inviting me for this webinar. Can I share the slides?
Yes, sir.
Yes, sir.
So, your students are attending seminar from September 7. former two speakers, Professor Mohan Isaac, discussed about how COVID-19 induces psychological stress in people, and also Dr. Unnikrishna discussed about detailing about plasma therapy, convalescent plasma therapy. So my area of research interest is on structural based drug discovery. So as you all know about very basic information about COVID-19, how it spreads, what are the symptoms and everything. So I'll be directly moving on the structural aspects of how the antibody discoveries and small molecule discoveries are done. So I have devised my talk outline like this. First, I will give you introduction about COVID-19. Then I'll give you an introduction about what is SARS-CoV-2 and how it replicates inside the cell. Then I'll be talking about neutralizing antibodies. What are neutralizing antibodies? How they are acting against SARS-CoV-2 with specific examples and case studies. Then I will discuss about how small drug discovery is done against SARS-CoV-2. I am working these aspects with my collaborators from Singapore. So, and also I'll be presenting other people's results also. So as you all know about what is coronavirus disease, I'll give you a brief introduction. Novel coronavirus disease pandemic since 2019, December is affecting the globe and imposing unprecedented challenges to the humanity. COVID-19 is specifically caused by a severe acute respiratory syndrome coronavirus, SARS-CoV-2. As of 14 December 2020, yesterday, there have been 2,89,18,900 confirmed cases of COVID-19, including 9,22,252 deaths reported to WHO globally. You can track this information in the WHO website. So, in addition to the supportive care, now scientists all over the world are concentrating on three major strategies to develop therapeutic against COVID-19 pandemic. One is vaccine, second one is the neutralizing antibodies, and the third one is the antiviral drugs. Compared to previous outbreaks of coronavirus such as SARS-CoV-1 and MERS, Middle East Respiratory Syndrome coronavirus, okay, SARS-CoV-2 is much more transmissible. It is transmissible to human to human. Large portion of the people are infected remain asymptomatic. So it makes a problem to have an effective containment and mitigation. The SARS-CoV-2 continues to spread slow with a little sign of slowing down, and the cases are increasing. So it urges for the rapid development of therapeutic and prophylactic treatments. What is SARS-CoV-2? SARS-CoV-2 is an enveloped positive sense RNA virus. It's a diameter of approximately 125 nanometer, characterized by club-like spikes. That's why it's called corona. That's why we call coronavirus. And it has a unique replication strategies. So it belongs to the beta coronavirus genus and shares similarity with the SARS-CoV-1 and also sharing similarity with the Middle East Respiratory Syndrome MERS virus and fat and pangolin coronaviruses. So basically, SARS-CoV-2 virus contains mainly four structural proteins. One is a spike protein, another one is the membrane protein, you can see in the picture, and the third one is the envelope protein, and the fourth one is the nucleocapsid. So these proteins are majorly encoded in the 3' end of the viral genome. You know, viral genome starts from 5' to 3' end. So these are coded in the 3' end of the genome. So the role of this protein, S protein, is to help the virus to attach to the host cell receptor. In the human case, it is Ac2 receptor. The M protein plays important role in envelope formation and a viral entry. And the E protein is highly divergent. It has ion channel activity and also facilitates assembly, budding, and envelope formation. The N protein has a multifunctional role, and it enhances the efficiency of viral transcription assembly. So this is the picture I simplified. This is the simplified replication cycle. Here you can see that the genome starts from 5' UTR to 3' UTR. Totally around 29 open reading frames are there. From the ORF1AB, most of the structural proteins are synthesized. So, and also apart from structural proteins, there are several non-structural proteins. NSPs are also translated. So S protein basically attaches to the receptors bound in the host cell membrane, like Ac2 and TMPRSS2. So it attaches to this receptor. This attachment facilitates the entry of the virus. Then, once the virus entered into the cell, the RNA genome is translated by the host ribosomes, and it produces several polyproteins which is needed for the assembly of the virus. These polyproteins were cleaved by several proteases. Two of them are important ones. One is the PL-PRO, which PL-PRO, that's called papain-like PRO. Another one is cysteine-like protease, that is M-PRO. M-PRO is also called a main protease. Then there is one more enzyme, RDRP, RNA Dependent Rna Polymerase, which involved in a RNA replication of the subgenomic RNAs. And these subgenomic RNAs move into the endoplasmic reticulum and Golgi intermediate compartment and get assembled and packaged, then released into the released outside the cell through exocytosis. Hopefully, most of the people know this process. So, for the effective way of designing drugs, for the rational design of drugs, so first we have to identify important proteins in the life cycle of the virus. So here, these 4 proteins play critical role in the replication of the virus life cycle. One is S protein, which is mediates the entry of the virus, and the second one is PL-PRO and M-PRO. These are enzymes; they process the polyproteins. Okay, and the third one is RDRP, which is the RNA dependent RNA polymerase, which is the central machinery of the replication machinery. So, if we are able to block any one of the sites, we could block the replication of the SARS-CoV-2. This is the logic behind why we are studying this life cycle. So first I will go one by one, and also okay. What is spike glycoprotein? SARS-CoV-2 spike protein is extensively glycosylated. Here you can see that there is 2 subunits, S1 and S2. S1 is in gray color and S2 dark gray color. And also you can see that the oligosaccharides, and the percentage of oligosaccharides surrounded by the along the protein surface. What is the use of this glycosylation? Viruses generally use this glycosylation to hide their viral proteins from the host immune system. Previous examples were like HIV virus, Ebola virus, and influenza viruses. They also use this kind of glycosylation. They seal the proteins, especially the envelope proteins, by glycosylation. This is called immune mimicry. So by this, they could escape the host immune system. So compared to S1 and S2 subunit is conserved among the coronavirus family. So that's why this S2 subunit is considered as an important target for developing drugs. So Ac2 binding site is the protein where Ac2 receptor is present in the host cell. So when the spike protein binds to the host cell, the Ac2 binding sites bind to the receptor. So this is the blue colored regions of the Ac2 binding sites. The first picture you can see the top view. When you rotate 90 degrees, you can see the side view of the Ac2 binding site. How SARS-CoV-2 virus attachment to the cell entry happens? I briefly explained. First, SARS-CoV-2 binds to the ACE receptor. Then And this AC receptor is cleaved by TMP-RSS2. It's a protease. It's due to a proteolytic effect cleaved by this enzyme. And this will facilitate the entry of the coronavirus inside the cell. There is one drug called nafamostat mesylate. The drug used to treat acute pancreatitis. This effectively blocks the entry of this virus. It can prevent the fusion of the envelope of the virus to the host cell membrane. This is the first step in the entry of the SARS-CoV-2. If you go further detail how this mechanism happens, you can see that in the gray color, the top portion is the host membrane and the bottom portion is the viral membrane. So from the viral membrane, the spike protein is protruded. So spike protein attaches to the Ac2 receptor. Okay. Once it attaches to the Ac2 receptor, then proteolytic activation happens. As a result, what happens is the S1 subunit, which is in the white color, which is the top portion of the spike protein, it is a trimer. This will be cleaved. Once it's cleaved, what happens, the next step is the S1 subunit there are regions which can facilitate the fusion of the viral and the host membranes. So setting of the S1 subunit triggers the fusion machinery. Once the S1 subunit is shed out of the spike protein, then there will be subsequent conformational changes which allows the S-glycoprotein to fuse to the viral membrane. As a result, the virus and the host cell fuses together. So we could exploit this mechanism using neutralizing antibodies to treat SARS-CoV-2. Neutralizing antibodies, you all know about antibodies. Antibodies have light chains and heavy chains. There are 5 different types of antibodies. These are the basic information we have studied in BSc as well as in MSc. So in the fight of SARS-CoV-2, antibodies are the key tool, both as a potential therapeutic and also it could guide the vaccine development. We can see how it helps in later slides. So recently, several encouraging preliminary results have been obtained by researchers. Okay, and so the molecular understanding of how these neutralizing antibodies interacts with the SARS-CoV-2. If you understand how these antibodies interacting with the SARS-CoV-2, then we can rationally design. That is, even computationally we can design. We can also screen drugs to fight against this virus. So the most important part is we have to understand how these antibodies interacting with the SARS-CoV-2. Before understanding, we should know how this humoral immune response happens against SARS-CoV-2 in our body. First, the SARS-CoV-2 enters into the host cell via interaction, as I told you earlier, via interaction with the spike protein and the Ac2 receptor. Then, following replication, the viruses are released into the host cells. A subset of viruses will be presented to the antigen presenting cells like macrophages and the dendritic cells, and they will be fragmented. These fragmented SARS-CoV-2 antigens will be presented to the T helper cells. These T helper cells will interact with activate the B cells. These B cells are plasma or memory B cells. They secrete antibodies, okay, for the high affinity binding to the receptor. Then these antibodies react back to the viruses and neutralize these viruses. Okay, this is what the basic mechanism happens when there is a viral entry into our body. So what are the potential targets of neutralizing antibodies against SARS-CoV-2? As I explained to you earlier, this SARS-CoV-2 envelope is rich of spike protein, which is S protein. Here it is in a green color, you can see. And it has 2 domains. One is called N-terminal domain, another one is called, actually it has 2 subunits, one is S1 and S2. And the S1 subunit has 2 domains, one is receptor binding domain and the N-terminal domain. So when a SARS-CoV-2 enters to the cell, mainly the receptor binding domain interact with the Ac2 receptor, viral receptor, and the N-terminal domain will be adjacent to the receptor binding domain. So this mechanism I explained earlier. where when the SARS virus enters into the cell, then there is attachment of S protein and Ac2 receptor happens, then cleavage of S1 subunit happens, then it forms a 6 helix bundle. This 6 helix bundle helps to fuse the viral and the host membrane. So several type of antibodies like monoclonal antibodies, FAV, single chain FV and HC antibodies and the nanobodies, and all these kind of antibodies can be designed or isolated from the for example, earlier lecture you have heard about convalescent plasma therapy, where antibodies can be isolated from the infected patients from the after 14 days. Then these antibodies can be screened, and we could find the antibodies which neutralize these viruses, and that can be used. So basically, these antibodies binds to the S1 subunit. Most of the time they bind to the receptor binding domain, and there are also some they target S2 portion of the antibodies also. So there is a group, Banas et al. They have identified, they have crystallized the structure of, there is a monoclonal antibody called the C105 bound to the SARS-CoV-2 primers. The gray ones are the spike protein. The spike protein, when they crystallize, they identified two conformations. One is state 1, where the RBDs, the receptor binding domains, which is shown in a red color, they are in the up position, okay, because this is a trimer. One is in the down position. In the state 2, there are all the 3 receptor binding domains are in the up position. So that you can see in the figure A. And figure B, you can see that the antibody C105, how it binds to the receptor. It mainly binds to the receptor binding domain of the S1 subunit. And also in the second picture, you can see that these loops are arranged around this receptor binding domain, CDRH1 and CDRH2, H3, CDRL. These regions binds. And here, in the surface representation, in the figure C, the dark green is the region where the heavy chain epitope, that means the heavy chain binds, and the light green is the region where the light chain binds. And why it is interesting? Because this is the same region where the Ac2 receptor also binds. So what happens is basically this antibody prevent the interaction of Ac2 receptor interactions. When the virus enters into the cell, this to enter into the cell, this spike protein has to be attached to the Ac2 receptor. So if you have an antibody which would block this interaction, you can minimize the infection or you can control the infection. Then there is a second series of antibodies that's called the Bd23 Fm antibody. This group have crystallized these antibodies. What they have found is these antibodies also interact with the receptor binding domain. Interestingly, there is a N165 residue, which is a glycan that also involved in participating in this interaction. So here only one receptor binding domain is in up conformation, other two are in the down position when they isolated this receptor. In the figure C, you can see that they have superimposed the Ac2, which is in the dark brown color, with the antibodies and the receptor binding domain. Here you can see that some of the Ac2 binding sites are overlapping with the Bd23 antibody. So the mechanism of this antibody, basically, it prevents the interaction of Ac2. It competes with the Ac2 binding. Then people have identified from convalescent plasma patients therapy, that's infected people. They isolated several antibodies and they characterized the antibodies. And there are several series of antibodies here. There is one is called the CC12.1 and CC12.3 and P38. So although they are different antibodies, but they share the interaction spots are similar. For example, in the right side picture, you can see that The CDR-H1 region is quite similar. The interactions are quite similar. That the bottom one is the antibody and the top one is the receptor. So and also, so only there is a major significant differences found in the CDR-H3 region. So what it means that these regions can be shared between the antibodies because they show a similar mode of interactions. What is the use of that? This is like when you have a targeting different regions of the epitopes or different regions of the receptor, then you can make a cocktails like we can use several antibodies for the treatment of SARS-CoV-2. That's why people are searching for the mode of interaction of antibodies. Then later people have identified there are some antibodies, they interact with the N-terminal domain, which is in the brown color, which is highlighted in the brown color. So one monoclonal antibody called A4A8, which exhibited high neutralization potency against pseudotyped SARS-CoV-2 viruses. And this antibody, it's bind to the N-terminal domain. It does not bind to the RBD domain. So far we have seen most of the antibodies bind to the receptor binding domain, but this antibody binds to the N-terminal domain. So, and these antibodies stabilized by mostly of hydrophilic interactions. The below figure you can see that the interface between N-terminal domain and the antibody, monoclonal antibody 4A8. So these antibodies can be used in the cocktail therapies. Like you can mix the antibodies for the treatment of SARS-CoV-2. People are doing some clinical trials using antibodies also. So far I have discussed about antibodies. Next I'll be discussing about structure-based drug discovery. Structure-based, if you remember the life cycle of the SARS coronaviruses, there are 4 stages which are important. One is viral entry, then the processing of the polyproteins, then by PL-Pro and the M-Pro, and the transcription of the transcription by the RNA Dependent Rna Polymerase. Okay, so there are currently no effective drugs or vaccines, and one of the strategies of treating COVID is to using the repurposed drug. Finding a new drugs normally takes a longer time because of toxicity and other side effects and everything. We have to evaluate. Minimum it takes 10 to 20 years, but due to the current urgency, it's very difficult to start from the scratch. Okay, so people are trying to use a repurposed drug. What does repurposed drug means? The drugs which are already approved for treating some other disease, which can be used in the COVID-19 treatment, because most of the drugs, they have crossed the safety screenings with the other previous clinical trials. So they are very less detrimental. So people are trying to identify repurposing drugs. So for 4 stages, they are trying to use these drugs. One is at the viral attachment and entry, and then the viral proteolysis, then the viral replication, then the host cytokine response. Scientists also exploring using different strategies like using artificial intelligence-based simulations, using supercomputing facilities, and also using some small molecule computation, docking, and structure-based drug discovery approaches. How it is done? Here I'm trying to explain with a few examples. So earlier we have shown, I have shown that with the S protein Ac2 attachment, we can design antibodies to understand it. We can also design drugs to inhibit this interaction. For example, the RBD of S1 binds to the host cell surface receptor of the Ac2. In the figure B, I have shown the Ac2 receptor, where you have the RBD, 2 RBD domains. Okay, and the figure A, you can see the spike protein, which is a homotrimer. So this spike protein binds to the RBD domain of the Ac2 receptor. Then there is a sequence called FP, which is a fusion protein. This fusion protein involved in the it acts as a fusogenic element when this virus attaches to the host cell membrane. So it forms a 6-helix bundle, and this bundle is facilitates the fusion of the virus. And there are two regions around this, following the fusion peptide. They are called heptad regions, that HR1 and HR2 regions. Okay, and this region can also be targeted for designing drugs. And if you remember, I have also told during the initial attachment, there is some kind of proteolytic cleavage is happening by TMPRS2. TMPRS2 binds to the S2 subsite. I have shown in the figure D. So this is a TMPRS2 structure is not available. So we have homology, did a homology model, and we have shown that this region is highly rich of acidic residues. So you can see that there is a red spot in this region, which binds to the S2 prime site. And also there is a cathepsin. This is also a protease. This region also highly negative, and it binds between the S1 and the S2 site. There are several drugs. They bind to bind to the spike protein. The scamostat and nafamostat and EST, MDL21870, and the imatinib. So chemostat and nafamostat, they inhibit S protein initiated fusion. They bind between the S protein and Ac2 and inhibit the fusion. And EST and MDL inhibitor, they inhibit the cathepsin L activity. They bind to the cathepsin L and they inhibit the cathepsin activity. Then the imatinib, you might have heard this. This is a Abelson tyrosine kinase inhibitor, which is used in cancer treatment. And this imatinib was found to inhibit the entry fusion. It can also inhibit the entry and fusion of the SARS-CoV-2. Then the next protein, which is important for SARS-CoV-2 life cycle, is 3C-like protein. It is also called MPRO. MPRO means main protease. So the major role of this enzyme is to process the polyproteins by proteolytic activity. So this proteolytic activity helps for the maturation of the virus. So SARS-CoV-2 improve have a 90% sequence identity with the SARS-CoV-1, and this improve has 3 domains: domain 1 and domain 2 and domain 3. So it is a dimer. Okay. So this homodimerization of these protamers, 2 protamers has to dimerize to be active as an enzyme. And this domain 3 helps in this dimerization process through a salt bridge interaction between glutamate and arginine. So the active sites of the MPRO are highly conserved, composing of 4 subsites. Here I have zoomed the 4 subsites: S1, S2, S4, and S1 prime, S1, S2, S4. So this is the active site where the substrate is processed. So this then you can see that there is a glutamate 166 in the each protamer. This glutamate 166 binds to the other protamer. Okay, and activates. It creates a pocket, S1 pocket for substrate binding. So the inhibitor is mainly developed based on the substrate it is processing. So the inhibitors are peptidomimetics. They mimic the substrates. Okay, and so they can form this active site can form a small scaffold for inhibiting MPRO. The people have identified 2 small molecule inhibitors, which is called 11A and 13B. And they are basically leucine phenylalanine dipeptides. So they have created a leucine phenylalanine dipeptides. The mode of action of this dipeptide is they covalently block the SARS-CoV-2 MPRO. They create a covalent bond with the cysteine residues in the active site, cysteine 145 in the active site, and inhibit the activity of the M-Pro. What will happen is when you inhibit the activity of M-Pro, it cannot process the polyprotein. As a result, the virus cannot mature. The virion particles cannot form. Then there is another drug, which is a drug we are interested. We are working on that. This is called Baicaling. It is a Chinese traditional medicine. It's a natural product. This natural product, it does not form a covalent bond like other inhibitors, but what it does is it occupies the substrate binding site, and it does not allow the substrate to enter. So it blocks the substrates approaching the active site. Okay, and it is blocking the catalytic cysteine 145. Then the next protease is the papain-like protease. This is a PL-Pro. PL-Pro is responsible for cleavages of N-terminus of the polyproteins. It cleaves the N-terminus of the replicase polyproteins. The structure of PL-Pro consists of 4 domains. One is called canonical right-handed thumb domain, and a palm domain, and a finger domain, and an N-terminus picotin-like domain. Here you can see I have colored in different colors. Yellow, green, blue, and dark blue. So these colors represent each domain. There are 2 distinct picotin binding sites that's called HUB1 and HUB2. Are found between the palm and the finger domain, as well as the ridge of the thumb domain. And there is a it is like the mechanism is like acid-base catalysis mechanism, where you have a conserved active site, a catalytic trade of cysteine 111 in the thumb domain, and histidine 272, histidine 272 in the palm domain, and aspartate 286. So these forms a catalytic triad, and in between the catalytic triad there is a zinc ion, which plays an important role in the structural integrity of this protease and also involved in the catalytic mechanism of this enzyme. So basically, this papain-like protease is a homotrimer. It forms a homotrimer. You can see in the right side of the figure there, these 3 subunits, they form a homotrimer and they process these polyproteins. So there are some PL-Pro inhibitors, people are trying to inhibit these enzymes. People have designed, there are 2 small molecule inhibitors called the VAR250 and VAR251. These small molecule inhibitors, they make a covalent bond with the cysteine 111 in the catalytic triad. As a result, it will prevent the substrate processing. Then there is also, like earlier we have seen in the case of M-Pro, there is non-covalent inhibitors, and people have also identified some non-covalent inhibitors like 15G and 3K. Okay, these are non-covalent inhibitors. They have a benzamide scaffold. They are based on the active site scaffold. They have designed this inhibitor, and these have good inhibitory activity. So what happens is that when these inhibitors, when they bind to the active site, it makes induced conformational changes. So as a result, the substrate cannot enter and it cannot be processed. So compared to VAR250 and VAR251, which covalently block the site, these 2 inhibitors non-covalently block the site and prevent the substrate approaching the catalytic site. In this way, they prevent the processing of the proteins. Then the most important enzyme and the most successful and is RNA Dependent Rna Polymerase. So currently, people are using this RNA Dependent Rna Polymerase inhibitor for treatment of COVID, emergency treatment of COVID-19. So how the mechanism happens is that RDRP is the central agent of the viral replication machinery. So RDRP also has several domains, N-terminal beta hairpin domains, then N-terminal domain adapting nidovirus RDRP associated nucleotide transferring architecture, shortly known as beta-NIRN. Then you have an interface domain, then canonical domain of right-hand structure. So you can see the coloring scheme. The coloring scheme represents the top coloring scheme. Also, you can see the domains architecture. The catalytic site of RP consists of A to G motif. Okay, A to D motif forms from the in the palm catalytic active center, and motif FG from the finger subdomain from F and G. Bind to the, this is the region where the template RNA binds. You can see that there is I have labeled in a dark blue color and also light blue. So template RNA binds in this cavity. This is the catalytic site. And motif F is involved in binding to the primer strand, RNA strand, and stabilize the incoming nucleotide substrate from chain proliferation. So it starts the chain proliferation. During the elongation phase, the nucleotide transfer reaction will be repeated with association of subsequent NTPs to generate a complementary RNA product. So the product hybrid will be out through this way, where you can see that there is a arrow which is marked, so the product hybrid will exist. So the second figure is the top angle, and the third figure is the 90-degree rotated angle. So the product is coming out of this. The most famous drug is remdesivir. It is RDRP inhibitor. So remdesivir is a prodrug of adenosine analog. That is, it is a monophosphate. Okay, then due to the host activity, it will be phosphorylated. It forms a primary triphosphate form. Triphosphate form is the more potent form of inhibition. So ATP is a substrate. So this RTP shows a similar binding to mode like ATP, but RTP is an analog. Okay, and there is a substituent and the 1' CN substituent. So what happens is here is that this CN substituent makes a steric repulsion with the residue in the active RDRP enzyme 861. So as a result, what happens? There is a distortion of RNA translocation in the I+4 positions. So the basic mode of inhibition mechanism is called non-obligate RNA chain termination. You know the RNA chain termination happens from 5' into 3' OH group. Generally, this analog stops at will be modified in the 3' OH. In the obligate mechanism, the incoming nucleotide cannot be able to order in the 3' OH positions. Whereas here, what happens is due to this substitution at the 1' CN substituent position, there is a distortion happening in the serine A61. So as a result, there is a steric repulsion which distorts the RNA transcription. Okay, and there is people have developed a orally bioavailable analog, which is called EADD2801, which is have a similar motif like RTP. And the special about this is it makes a 3 hydrogen bond in the with the base pairs. So this results in a higher potential than RTP against SARS-CoV-2 replication. So what is the use of understanding this is if you understand the molecular interaction between how these drugs as well as the antibodies react with the host membrane, and if you know the structural aspects, and if you are interested in the structure-based drug discovery, you can devise your own inhibitors to control SARS-CoV-2 replication. So as a summary, this COVID-19 is a once-in-a-century blow to the global community. We are all experiencing the stress associated with COVID-19. It is overwhelming and not sustainable. And although we have taken rigorous containment quarantine efforts, number of confirmed cases increases daily. Up to now there is no first-line vaccines or treatment for COVID-19, so that we are collectively gathering insights from the viral life cycle and how this disease progressing mechanisms. And so all over the world, the researchers are trying to develop drugs, vaccines, as well as antibodies to fight against SARS-CoV-2. So by combining these structural data and target proteins and also using deep learning algorithms and computational tools, and with the help of medicinal chemistry, hope we will be finding a therapeutic solution soon. Thank you. Thank you. I have given the references to further understand. The first reference is our review article we have published in Ang & Academy. So most of the content of the talk is from this review article, small molecule based drug discovery. And the antibodies part, we are writing a review article. We'll be sending you soon. I have given 9 references. So you can go through these references. If you couldn't find a PDF, you can write to me. I can send those files. If anybody interested, you are welcome. Once again, thank you for the management, principal, and staffs, especially Phoebe Kichan for inviting me to present this seminar. Thank you. If any questions,
Thank you, sir, for your guiding and academic presentation. Now the session is open for interaction. Participants, please put your queries in the chat box.
how can I see the questions?
So when you have questions, I'll read them out to you.
Okay, thank you.
So we have a question.
Okay.
Is there any side effects for Bacalin? Is it FDA approved drug?
No. It is Chinese traditional medicine. People are in China. People are using like a traditional medicine. How we are using our traditional medicine like tulasi or some kind of Ayurvedic plants also we use without. So like that, it is Bacalin is it's a Chinese natural. It's a plant. From the plant, people have identified this Bacalin have some activity to control. Initially, it was identified to control dengue virus. People traditionally use for controlling dengue virus in China. So our lab means my collaborator labs were interested in studying this molecule. So now we are trying to modify this Bacalin and try to see how it. Some of the preliminary results shows it's promising. So we are trying to. Basically, I am a computational drug discovery. My PhD is on computational biophysics. So we are doing some computational docking and the simulation kind of analysis to see how these drugs can be modified and how we can make more effective for increasing the binding to the SARS-CoV-2 proteins.
Okay, sir. We have another question. It's from Licinta. What are the factors considered in clinical trials of investigating COVID vaccine? And what about the efficiency of these trials?
Actually, there are 3 trials, phase 1, phase 2, and phase 3 clinical trials, right? So there are different kind of vaccines available. So for example, there are vaccines which most of us know about attenuated vaccine, right? Then we know about recently there is a company called Moderna. They are trying to develop mRNA-based vaccines. Okay, and and also there are subunit vaccines like subunits like I talked about the spike protein, right? So so each subunits you can develop a vaccine. At the same time, you can also take the mRNA of the coding of those subunits, and you can also develop a vaccine. So recently, the Oxford vaccine what they have developed in the phase 3 clinical trial they started. So after trials only we will know what kind of side effects are coming.
Okay, sir. We have another question from Anisa Miss. What are the main challenges in raising antibodies against a viral particle? Can SAR studies complement these challenges?
These viruses they tend to undergo a mechanism called mutational escape. So, for example, you are developing an antibody against a particular epitope, so a particular region of the protein. What happens is during usage of antibodies, sometimes what will happen is this virus can undergo mutation in those regions, and the antibodies might lose its effectiveness. That's why people are trying to nowadays they are trying to develop what you call cocktail antibodies. So suppose if the virus becomes ineffective for one particular antibody, so the other antibody can neutralize. So people are trying different other mechanisms like recently people are trying to engineer the Ac2 receptor, where they increase the affinity of the Ac2 receptor. So you can produce a soluble Ac2 receptor. So when you inject this soluble Ac2 receptor, it will bind to the, so the virus binds to the soluble one, not to the attached one. So as a result, you can also you can neutralize. The mutation is the main problem when you are trying to develop antibodies or vaccines. So what we generally do is we do in computational we do called conservation mapping. So each residues mutational conservation mapping. So each residues we substitute with different amino acids and see how these amino acids changes the structure, also changes the binding affinity, and all these things do a computational calculation also. So in the case of there is a called in case of small molecules there is a technique called fragment screening. So small fragments can be screened, then later these fragments can be assembled together to form an effective drug molecule for binding to the proteins. So since nowadays the computational problem is so fast, so screening per day we can screen around 10,000 to 20,000 molecules, and so especially during the COVID period, it is going in a tremendous fast manner.
Let us have a look on the YouTube chat box as well. Priya Miss, please.
We have not received any questions in the YouTube chat box. Thank you so much, sir, for your patience in answering all the questions.
Okay, thank you so much. Okay. So if you have any further questions, if you have any further questions, if you come later, some questions or something, you can write to Phoebe Madam. Then Phoebe Madam can transfer this question to me. I could try to answer. Or any one of your staffs, you can raise, then I could answer this. Thank you.
I believe that our speaker has cleared almost all the queries raised by the participants. Sir, would you like to share anything more?
Yeah, basically I started my career as a teacher. Phoebe introduced me as a JJ College. what I observed is that both BSc and MSc level students mostly concentrate on studies. They are studying, but there are nowadays earlier people don't have much accessibility for the reagents or doing experiments. Nowadays there are a lot of computational. Everybody has the computers. There are a lot of ways you can do research. Like you can go to the protein data bank and download the proteins and you can visualize the structure. And you can also, there are a lot of tutorials available like docking and as well as, so you can help this drug discovery process. If you have some ideas, like you have some exciting ideas other than what is currently undergoing, you can talk to the researchers, or you can check which are the labs they are doing this kind of research. You can talk to them. You can indirectly help them. It is indirectly help them. Later you can apply for your PhD or postdoc. It will be helpful. So this is just a suggestion. So you can. So there is no need for a lab to do research. That's what I basically want to mention. Nowadays, computational powers are so much. With the laptop, you can do. See, most of the work what we have done is with the personal computer and the supercomputing facilities. So especially these MSc people could try that. There are a lot of tutorials available in each software and the YouTube as well as you can self-learn and you can talk to researchers if you have a doubt and everything. So that's just a suggestion.
Thank you, sir. The feedback link is now available in the chat box, so participants make sure that it is noted. It is time to wind up our session. I invite Srimati Gia Paul, Assistant Professor of the Department of Biosciences, to give the vote of thanks.
Thank you, Ashley. Thank you, Dr. Kathireshan Natrajan, for your informative talk. I, on behalf of the Biosciences Department and the entire fraternity of Sri Shankara College, Kaladi, and on my behalf extend a very hearty vote of thanks to you for sharing with us your findings and opinions today. Thank you, sir, for your efforts towards the speech titled on Neutralizing Antibodies and Molecules Drug Discovery against SARS. The discovery and development of virus neutralizing monoclonal antibodies could be one approach to treat or prevent infection by this coronavirus. Hope all of you have benefited through the webinar. It's audience like you that make presentations for your unwavering attention throughout this presentation. I am happy to thank Sri S. Prasad for his firm support in this event. Finally, I would like to take this opportunity to place on record my hearty thanks to my colleagues and the management for the perfect logistics support and guidance extended for the smooth conduct of Biogen 2020. Thank you all.
Thank you.
Thank you, ma'am. Our next technical session of this webinar series is tomorrow, 16th September 2020. The link for the next session will be sent to your email ID before the start of the session. Once again, I thank everyone for your valuable presence. Dear participants, we once again remind you all to fill up the feedback form as early as possible before leaving. The link will be active only for 1 hour after the session. It's the time to disperse. See you in the next session on 16th September. Thank you and have a great day.
Thank you.